Membrane transport and facilitated diffusion of the phospholipid bilayer cell membrane. All right, active transport. So, active transport is a molecule that is going to require energy. So, it's going to move usually solutes against a concentration gradient. It's the most common thing that active transport is used for. These are things like pumps, and then perhaps you remember endo and exocytosis, and then co-transport, which is used to uses a lot of energy to move particles across the membrane. So active transport is going to require energy, and so like what is energy? And that is ATP. Adenosine triphosphate is the energy molecule for cells. It is the source of energy that cells use and process to carry out their metabolism. And so ATP can transfer these terminal phosphate groups on and off of the molecule, and that happens inside of the transport protein. And that changes the shape of the transport protein to better move the substance. In terms of protein shapes or this molecule shape, we call that a conformational change. So the molecule itself isn't changing, it's changing its conformation. It's almost like it's flexing. But essentially, it's going to lose. This guy right here is going to leave. It'll be ADP, adenosine diphosphate. Pumps, they maintain cell membranes potential. Membrane potential is this unequal concentrations of ions across a membrane, and it results in an electrical charge. It ultimately an electrochemical gradient, if you will. Remember, some ions are positively charged and some of them are negatively charged. So you can create this positive negative interaction by moving ions to one side or another. And this is going to require energy, right? Because usually we want to move this like against a gradient. So things that do this are called pumps. The cytoplasm is relatively like this negative ion-filled fluid in comparison to the extracellular fluid that's on the outside of the cell. And so just for biological functioning, there is already a electrochemical gradient between the internal component of the cell and the external extracellular fluid. And it's almost like there's a stored energy option gradient that's there. So let's just talk about a couple of examples of pumps. An electrogenic pump is actually a protein that's going to generate voltage across a membrane. And when it does this, it is going to store that energy source so it can use it later. One of the ones you'll hear about a lot is a sodium-potassium pump. This really only takes place in animal cells. That is because we really need to regulate our sodium and potassium ions in and out of our cell. For every 3 Na sodium plus ions that get pumped out of the cell, we need to pump 2 potassium ions into the cell. So there's this resulting positive 1 net charge in the extracellular fluid. And that's where we're getting this pumps that are going to require energy, because it's pumping it against a gradient, basically leaving a positive charge on the outside of the cell. It's really quite fascinating how it works. Animal cells, they have a relatively high concentration of potassium inside of them. I mean, in comparison to like what's on the outside of the cell. And so they're going to have a relatively like low concentration of sodium on the inside compared to the outside of the cell. So ATP is going to be used and it's going to phosphorylate this protein and cause the protein to do that conformational change where it changes its secondary, tertiary, quaternary structure. It's not physically changing the protein, it's just like flexing its structure on the inside. But this time it gets to use ATP to do it. And the shape of this protein has an affinity and an electrochemical attraction for sodium. So it definitely favors sodium. All right, so the pump likes to take these sodium molecules, and pump them out. Every time it pumps 3 of those sodium ions out, it'll take 2 potassium ions in. So this particular pump, we hear about it a lot. Think about it as it's moving sodium out and it's uptaking or moving potassium into the cell. And it's a special pump. It is definitely responsible for moving sodium out of the cytoplasm. But it can't do that without potassium. And so this is definitely something that we will investigate further in class. Once it pumps the three sodiums out, the 2 potassiums fill that space, and the potassiums bind to this protein, and this phosphate group inside of that protein is actually released. And so the new shape of the protein causes an affinity now towards the potassium ion. Then it's going to lose that phosphate group, causing the protein to open and return to its original shape, and the pump will start again. The proton pump in cells is another good example of an active transport system. So this is an integral an integral membrane protein that builds up a proton gradient across the membrane. And this is really used by plants and, of course, a lot of fungus and bacteria. And so it's pumping these protons, also known as H+, out of the cell. Now, if you think about it, why would it want to pump H+ out of the cell? Probably has something to do with that. Co-transport, active transport, is a coupling of favorable movement of one substance with an unfavorable movement of another substance. So this process definitely uses energy. Remember that stored electrochemical energy gradient that I talked about that's generated by pumps? Well, it's now going to use that to move substances against a concentration gradient. I suppose I should also clarify what this movement is. If we call it favorable, then it's moving in a downhill diffusion way. And if we call it unfavorable, then it's moving uphill transport. So these terms are, counterintuitive, maybe, because if something's favorable, you think it's moving. Anyway, if it's favorable, it's moving downhill in diffusion. And if it's unfavorable, it's moving uphill transport. Plants use this co-transport for sugars and amino acids. An example would be that H+ co-transporter on sucrose. So sucrose can travel into a plant cell against its own concentration gradient, but only if it's coupled with this proton. It's kind of almost like the tag it uses to, kind of take a ride. And so that proton is going to diffuse down its own electrochemical gradient and take with it sucrose. So this co-transport system and this proton gradient, co-transport meaning two, they're cooperating. We have two different proteins here. Sucrose is not able to move across the membrane, and sucrose is actually synthesized inside of the cytoplasm, but it likes to cross membranes and go into adjacent cells. Because it's this relatively large polar compound, it's going to require these proteins in order to move it across a membrane. So the proton that's attached to sucrose is going to move in a passive fashion down its concentration gradient. So the pump is actually pumping ions, H+ proton, that's why it's called proton, because it's hydrogen. It's pumping it out of the cell. So when it does that, it creates a concentration gradient, and then the H+ that's attached to the sucrose molecule can then passively but assisted with this protein channel move into the cell. So essentially the co-transport proton system allows sucrose to move across this concentration gradient into other companion cells. But there are larger molecules than sucrose, right? So the transport of these very large molecules is done through a system of exocytosis to exit and endocytosis to enter. Exocytosis is the secretion of molecules via these vesicles, that fuse to the plasma membrane, and then these vesicles can then go ahead and fuse to the membrane, become part of the bilayer. Once they're fused, the contents of that vesicle are expelled into the extracellular fluid. Nerve cells do this really, really quickly and often. That's how they release these neurotransmitters. Endocytosis, the uptake of these molecules from vesicles, happens in the same fashion, where the vesicle wall itself will fuse to the plasma membrane and the cell will intake things. They can be pinocytosis, which is like a fluid movement, or phagocytosis, which is when it refers to solids. When the active transport endocytosis system is receptor mediated, it will take in very large things, larger than an ion, of course, so a virus, and also hormones and other proteins that can assist the cell internally. So this is best if I just show you a picture of each of these. So phagocytosis is when that cell is going to engulf a particle, and that will be later digested by lysosomes. So the cell surrounds that particle. It has this pseudopodia that just, like these guys right here, it's almost like little pseudopods. They will extend out. That's part of the cell membrane. It's extending out, and it will engulf these larger particles, food vacuoles, and bring them into the internal membrane, where lysosomes will eventually find them and digest it, the internal contents. When it uptakes extracellular fluid, that is far less specific, and it's far less picky about this. It's going to uptake anything that is extracellular on the external part of the cell, and take in those particles into the actual cell. And the protein coat helps kind of like mediate the transport of these molecules a little bit, but it is essentially an intake using vesicles that are more fluid in nature. And then these receptor mediated endocytosis vesicles, they're specific, and they uptake these molecules via a solute. It binds to those receptors on the plasma membrane, and this allows the cell to take up like large quantities of very specific things. When solutes bind to those receptors, they kind of cluster, and they coat the vesicle of sorts, and then once they coat it, they bring it into the internal part of the cell. And so this is actually a pretty good illustration here, where you see these are the receptors, and they are internally binding, and they take in this vesicle like this. And, these receptor mediated ones are just using receptor proteins on the outside of the plasma membrane to trigger or find these vesicles. And they're going to move pretty large things, like an entire virus or a hormone, which is a really large molecule compared to what we've been talking about. So I take a moment to kind of concept check. We've talked about a lot. Let's do a practice FRQ. You want to determine the effects of pH on the uptake of sucrose in tomato plant cells. So you're going to expose the cells to a sucrose solution at varying pH levels and record the data seen in the chart. So you did that and you recorded that data. So we want you to identify the optimal pH for the sucrose uptake, and then describe why the pH identified in the first part would lead you to increase in sucrose uptake. And this is pretty typical of an AP question, like, get part A first, and then for part B and part C, like use part A. And then finally, we want you to predict how a pH of 2, which we didn't use, would affect this sucrose uptake. Another way to concept checks would be to understand, like, perhaps you have a pathogen like bacteria and it invades the human body. So the immune system is altered. Once it's altered, white blood cells like macrophage, they're going to find this pathogen and engulf it through phagocytosis. Now, once it engulfs it through phagocytosis, remember we have those lysosomes to take it apart and digest it. So we want to describe the process of phagocytosis, and then also identify the most likely component of the plasma membrane that the immune system uses to recognize the pathogen, which we have talked about, but I did not point out that it was specific to the immune system.